So today I'm going to be talking about Virginia Man-Yee, who is a neuroscientist that helped discover the protein causes behind major brain diseases such as Alzheimer's and then Parkinson's. So she didn't begin on a normal science path because her mom wanted her to study piano at the Royal Academy of Music in London, but then she kind of just thought that music was not the right career choice for her, so she switched to chemistry and biochemistry. And then later, she earned her PhD at the University of California. Um, she kind of faced some obstacles from her family because they wanted her to be specifically a music major. So she stated that she often had to make her own path and then didn't really have a clear role model. One of the most interesting hobbies or facts I found about her is that she had a strong background in piano and that was actually supposed to be her future career, not to be a scientist. Something I admire about her is that she was brave enough to change the direction of her career path. And she didn't really start in science, but she became, I feel like, one of the most influential researchers within neurodegenerative disease. And I feel like I identify with this because she kind of just shows that you don't need a specific and perfect path to get to where you need to be. You kind of just need to have your own path. She works in neuroscience, specifically the molecular and cellular biology of neurodegenerative disease, where her labs kind of studies the abnormal proteins that are involved in the diseases like Alzheimer's, Parkinson's, amyotrophic lateral sclerosis, and then frontotemporal degeneration, where some of the big questions that they ask are which proteins are causing damage, how they spread through the brain, and then how that process can be stopped. One of the recent primary research articles that Lee and some of her colleagues had was in 2024 targeting endogenous tau in seeded tauopathy models inhibits tau spread. And this paper kind of studies whether an antibiotic called the mTau8 was able to reduce the buildup and spread of tau pathology within the cell and then mouse models. A good figure to explain this would be the culture neuron experiment, where the mouse neurons were exposed to Alzheimer's disease tau seeds, and then they were treated with either the mTau8 or a control antibody. With this figure, it shows how much abnormal tau accumulated afterwards. I feel like this is a strong figure because it connects the treatment to the amount of pathology. And then in the figure, you can see that the neurons treated with the mTau8 antibody show a lot less tau buildup compared to the control group. So this shows that targeting the cell's own tau protein is able to reduce the spread of tau pathology. And then this shows that tau could be a strong target for some of the future treatments of neurodegenerative diseases. The figure was kind of just asking whether targeting the neuron's own normal tau could reduce the formation of new tau aggregates after harmful tau seeds were introduced, or in other words, saying, could blocking the host tau protein slow the spread of tau pathology? The research used cultural mouse hippocampal neurons and exposed them to tau seeds derived from Alzheimer's disease brain samples. They treated the cells with either the mTau8 antibody or control antibody, and then they used immunostaining and biochemical assays to measure abnormal tau buildup, especially the insoluble aggregated form. For the experiment, in simpler terms, the scientists grew the mouse neurons in culture. They added the Alzheimer's-derived tau seeds, treated neurons with either the mTau8 or a control antibody, and then they later measured how much abnormal tau had built up inside the cells. One of the main controls was a nonspecific control antibody, so the researchers were able to compare it directly with the mTau8 within the same conditions, and then they also used cell counting stains to show the effect was not simply just because of killing neurons. And then they compared insoluble and soluble tau to focus on the disease-related aggregation. One of the main conclusions was that the mTau8 was able to reduce the tau pathology in the neuron model and in seeded mouse models, and this shows that antibodies do not only work by binding the incoming seeds, but also by targeting the neuron's own tau and then blocking the growth of new aggregates. So far, Lee helped identify some of the most important disease proteins in neurodegeneration. Some of the discoveries in TDP43 aggregates in ALS, frontotemporal dementia, distinct alpha-synuclein pathologies in Parkinson's disease, and then multiple system atrophy. But in a broader sense, just helped establish that abnormal protein aggregation is one of the major drivers of neurodegenerative disease. Some of our next steps are centered on understanding how the proteins like tau, alpha-synuclein, TDP43, and then APP behave in diseases, and then how that knowledge is able to be turned into therapies. These are my references, with this being my primary literature, and then some of these are my other sources used.